Good afternoon, everyone. This is Dwayne Schultes from Vital Transformation. I'm here at Zinn, and we're here to discuss our recent BMJ publication looking into new methods for comparative effectiveness harnessing real-world evidence. Sitting next to me is Dr. Amir Makadi. Hello, Amir.
Hello, Dwayne.
And we're joined on the phone remotely by Anya Scheel from NOMA. Anya, how are you doing?
Fine, thanks for inviting me, and welcome everybody.
We've just to everyone, we've had a bit of a lot of technical issues getting here. Anya's completely lost all internet in Norway. There was a huge storm, and we've had a ton of issues getting IT together here on our site. So it's been a bit of a scramble, but we're very glad to see all of you out there in the audience, and we will press on because I think everything now is under control. So we can hear Anya and everything's fine. So thank you very much for all of you who've registered. We have over 250 of you who've registered today. Again, thank you very much for your participation. It's very good considering it's August. Again, we do this webinar virtually. We've provided a bunch of dial-in phone numbers. Obviously, most of you are probably on the computer. If you are having issues, you can change from computer to phone. There are 24 international phone numbers. Please feel free to dial in if you're having connection issues, and you should be able then to talk with us, send us information, whatever your bandwidth should work well. The final half hour or so, depending on how long we go, will be open to question and answer. So please feel free to submit your questions. There's a little question bar on your there's a little Q&A bar on your menu bar there at the bottom. So please feel free to send us questions that come in, and we will answer them as we move through the webinar as we can. So the last half hour will be dedicated to answering your questions and answers. So to begin, this is a webinar discussing our recent study, what was published in the British Medical Journal Evidence-Based Medicine. It was funded from a grant from the Dutch government and the Zorgens Institute. And basically what we were looking at were CAR T therapies, and are they living up to their hype? How are they actually performing in real world using real world data? The publication is open access. All of you can download it. It's available online in PDF, and it's done very well. It ranked in the 90th percentile as far as impact. So we're very pleased with how it went. And again, Amir, thank you very much for your support on this.
It's a pleasure. Thank you too.
So the study itself, we're going to just dive in and we're going to talk about the results here. The study was funded by Zorgens Institute, the Zinn, as we mentioned, and we partnered with two institutions in the United States. We partnered with Vanderbilt University and Nashville Biosciences, and we did so because they have a very robust EHR system that is very accurate. Dr. Brian Rothman has been curating that system, and Leland Ekstrom, from the former partner from healthcare and McKinsey in the US, now runs Nash Biosciences, which is a commercialization arm. So it gave us really accurate data on CAR T, and we were able then to extract through the EHR patients that existed for several years. So even though the drugs have only been, the CAR T therapies have only been on the market since September of last year, we were actually able to construct quite a robust longitudinal pathway, which allowed us to get a pretty accurate assessment of what's going on. And in this sense, it was quite a unique opportunity, and that's why we partnered and sort of developed this methodology. So why this study? What were we trying to figure out? With the increasing use of new tools like FDA's breakthrough, as well as other accelerated pathways, it's really difficult for HTAs right now to make an accurate assessment of long-term value because generally these are extrapolated from smaller data sets. So as I mentioned, for 10 years now, VT has developed an EHR methodology where we extract real-world evidence from EHRs, and then we model them to actual cost data. So rather than using a proxy, we extrapolate. And so this study in acute lymphoblastic leukemia, ALL, the CD19 CAR T therapies, was really a proof of concept. Now, Anya and Amir, before we go forward, from your perspective, why is this such a challenge right now? With the idea that you're getting these, I mean, obviously we want to go earlier and earlier, but what's the challenge from an HTA perspective? I'll start with you, Amir.
We'll come back to it later as well in the slides, but just briefly summarized, we are getting evidence packages which are perhaps correctly, due to the potential cure and potential innovation of these therapies, is relatively limited, and we are forced to make a decision. We cannot postpone a decision and ask for evidence later on. And of course, the decisions we make have very long-term implications, as in once something is reimbursed, it's very difficult to remove it from the reimbursement list.
Sure.
So it's an early time point. We have to make a decision, and the consequences of that decision carry on a long time after that decision has been made. That's briefly summarized. Anya?
Yeah, Anya, from your perspective.
Yeah, I can only echo what Amir was already saying. The point is that HTAs have to be, to some extent, a bit risk avoiding, because obviously we have to make decisions in situations of high uncertainty. And the more limited the data, the sparser the information, the higher the risk that we make the wrong decision. And in that case, most HTAs will have as a default almost to rather say no than yes. And that is, I guess, where most people say, well, you should be neutral when it comes to risk taking, but most people will realize in everyday life, you do not take risks in a certain direction if you don't have to. And I think that is one of the underlying problems here.
Yeah, and one of the things we found in the common body of knowledge related to the CAR T therapies that had come out when we were preparing for this study was, a lot of the data had been extrapolated on the release by 6 months. So you had 6 months of actual hard outcomes and then 2 years of data presented in the application that had come in. And we thought, well, wait a minute, we might be able to do something a little differently, which is why this was just floated as a proof of concept. And actually, given the response to the BMJ article, it went pretty well. And Amir's going to talk a little bit more about that. And Anya, you're going to cover, there was a paper that came out from ICWIC discussing this problem in more detail, and you'll discuss that later, right?
Yes.
Fantastic. Okay. So with that, thank you very much. We're going to move on. So again, I mentioned that we did this study in acute lymphoblastic leukemia, ALL. Now, these are the two drugs that have recently come on the market from Gilead and Novartis. And again, I need to stress that the drugs we're looking at are not specifically for any indication. We were just looking at CD19 as a class. So we were just looking at the performance of the drugs as a class. So how does the current treatment pathway work? This is sort of a Markov type model. And essentially what you find is that, 80 to 85% of the adolescents who come in with this type of leukemia generally respond first line. In other words, they walk out healthy. And then 15% are non-responders, and a certain percentage of those are refractory, who just don't respond at all. And a certain percentage of those are partial responders. So what's generally been happening with those patients who are non-responders or refractory, they're generally getting bone marrow transplants, and that is the preferred pathway before CAR T. Now, the challenge there is, in order to get a bone marrow transplant, in order to have that window to have a bone marrow transplant, you need a couple months of clean remission. And with a lot of these non-responders, that's not possible. And so what we were wondering was, what's going on with CAR T? How is it actually being placed in the market? And what's going on with these non-responders and these partial responders? So what we developed here, we had a database of 2.8 million patients over 5 years that we extracted from, that had some type of ALL. So basically, we were looking for people under 25 years old who developed the symptoms for ALL, and we had a database to play with of 2.8 million patients. Now, from that 2.8 million patients, there was an N of all possible cases of 637. So you can see this is really, we're talking about quite a small population. And what we found was, from that 637, 529 responded first line. In other words, they walked out of the hospital. We saw no further treatment. So we assume that they, responded to treatment as they should, and that's about 83%, which is what we'd expect to see. And that left 108 patients that went second line. Now, of that 108, we had large data gaps, and 62 of them of more than 2 months. And in order to preserve the accuracy of the analysis, we decided to leave those out. We didn't want to bias the sample or, put anything in that wasn't certain. So we wanted to make sure that we only use certain patient data where we had a good, robust longitudinal pathway. And from that, we had 29 bone marrow transplants. That's the HCT patient pool over here on your left at the bottom. And then we had 14 CAR T patients, as you can see on the right. Now, we'd also segmented out blinatumomab, some of the late stage monoclonal antibodies. We actually see that as a precursor treatment used both for HCT and CAR T, sort of as something to sort of sustain a remission. So we decided to leave that out of the analysis as a whole. So what we're dealing with here is pretty small numbers, 29 and 14. So again, 29 and 14 out of 2.8 million. That just shows that, we're dealing with quite a small population. For those of you who aren't aware, in the US and Europe alone, they're looking at 700 patients a year in ALL. So it's a very, very targeted therapy, certainly an orphan. Now, when we talk about big data, well, here you go, folks. The ICD codes, we played with ICD codes. Those were extraction based. 500,000 of those. We had cost codes, 750,000 cost codes. We had 2.5 million drug codes. So you want to talk big data? There it is, ladies and gentlemen. That's what you get. Even with a small population, such as 600 and some odd patients out of 2.8 million records, you're still dealing with enormously large data sets. So what we did is we took those patients that were extracted and then we linked them using the HCPCS cost codes of Medicare and Medicaid. So we were actually able to get cost codes for everything, tongue depressors, aspirin, you name it. And then we looked primarily at the meds because those were the main cost drivers as well as the treatment pathway. And we were able to get a very accurate assessment, a sort of vanilla cost between the bone marrow transplants and the CAR T therapy to see what was going on as well as the outcomes. So one of the problems that you have with ICD codes is there is no "they're healthy here" code, "they're not healthy here" code. Those codes do not exist. They're just not there. So you have to develop proxies to figure out if someone is sick or not. So what we found was a very marked increase in the amount of total activity in the records. So these are ICD codes, drug codes, cost codes. When you amalgamated those and looked for spikes beyond 2 standard deviations of activity, which is what you see here in this chart, you see the spike. We found then that we would go in at those very points and look for a change in therapy regime that would trigger a relapse or indicate a relapse. It allowed us to a very accurate assessment, in fact, to be able to figure out using ICD codes alone and just using a quantitative analysis, a way to calculate relapse. And it actually turned out to be quite accurate. So what you see here in this patient, this is a patient who presented first line, had a relapse, and then was given a bone marrow transplant. You see second line, and then the patient relapsed off the bone marrow transplant and then was given CAR T. So this is what you're seeing here, and you can see how it aligns with the spike of activity. This is how we were very accurately able to determine relapse, which has been a big challenge. It's a novel methodology. It's something new that Dr. Daniel Gasol and I developed. It's mostly Dr. Daniel, actually. Thank you, Daniel, for your work on this. So we're very excited about this because this is a novel approach that we think could work and could also become an AI application. So what you're all waiting for, here you go, the Kaplan-Meier curves of CAR T to bone marrow transplant. And I'm sure some of you now are probably not happy, and I'm sorry, but this is what the data is. So what we see is the 3-year survival probability of bone marrow patients are about 68%, whereas the 3-year survival probability of CAR T patients in our patient population is about 50%, a little less. Now, granted, that's a very wide confidence interval, but it does bias to the downside. So we're confident with this and we'll stand by that data. It is reflective of what the practitioners have been seeing. Now, why is this? Why is this divergent from the studies that have been presented as evidence? Now, that became the big question. Obviously, when we saw that this is so different from what's been published in ICER that we figured that there was probably a treatment bias or something going on in selection. And so we dug into that. And we did, in fact, find a treatment bias. Now, this is a bit of a complex chart, so I'll try and walk y'all through it. The blue line, the blue descending line, represents the treatment previous to having either CAR T or bone marrow transplant. So that's the previous treatment. So this is what happened before they went to the second line advanced therapy. Okay, so what you see is that the CAR T patients, the patients who received the CAR T therapy, that's the chart on the left, have a much shorter overall response in their previous chemo treatment than the patients who received the bone marrow, the HCT patients on the right. As you can see, it's over twice as long in the response overall. Now, what this means and what we saw in the data and what we confirmed with the practitioners at Vanderbilt University was that the CAR T is being reserved for those patients who are non-responders or refractory, who they cannot give a bone marrow to. So basically the transplant units globally, and we've confirmed this with several other hospitals, are defaulting to bone marrow transplants before they give CAR T. Now, there's some reasons for this. There's some financial disincentives. The fact is right now hospitals in the US lose a lot of money by giving CAR T because they don't charge as much, and we'll show that later. But there's also a problem where it's a new therapy, you know bone marrow works, you have the systems in place, and so you're only giving CAR T to those patients where you know that they're not going to respond and you have no other option. Now, it does open up an option, and yes, that is an extra 1,000 days, and that's pretty good. As a matter of fact, it's very good. but it doesn't respond as well as the bone marrow patients. So what we're seeing here is a treatment bias and a selection bias in that we don't know how CAR T performs in the patients that aren't more difficult, aren't more challenging, because right now, those are the only patients who are getting it. Amir, do you have a reflection on that from your standpoint? From an HTA perspective, what does this mean to you when you see this data compared to what's been submitted to you in the applications?
Well, I mean, I think you greatly emphasize the most important points. If I would only add very briefly is that um, it becomes a question: what would happen if you move CAR T to earlier lines? Because we
Correct.
see that happening a lot in oncology, and the claims that come with these kinds of advanced therapies are also related to cure. So on the one side, we could be quite harsh and say, well, the cure only happens for a very small percentage or has not yet been fully represented. On the other hand, as you said, we do sit with the societal aspect of an extra 1,000 days is probably a lot more than the average 3 months extra survival
Yeah.
that you see for some other cancer products. So it's a very complicated picture, as you already have implied throughout the presentation.
Yeah, I should add that the practitioners we were working with at Vanderbilt said, as of to date, with the patients that they've given CAR T to, they do not see it as curative. Now, that's their opinion, and that's what they've seen in clinical practice, and that is verified in the data that we've seen. That's not to say it won't be curative if you get it to the less disease burden populations. But I think what's interesting is that if you read the studies, even now, particularly some of the studies that are coming online today, the refractory patients and non-responders are being excluded in the inclusion-exclusion criteria.
Absolutely, and that poses an even bigger problem for HTA agencies as well. And I think I don't know if we're going to touch on this in a following slide, but the idea that for some clinicians, CAR T therapies can actually be seen as a bridging therapy towards stem cell transplants. Now, that brings a lot of budget considerations along as well for HTA agencies, which we have not seen in the evidence base presented to us.
Yeah, I'm limiting the paper right now in the presentation to what's in the paper online. We did have one patient, and again, it's an N of 1, which is why we excluded it from the publication. But we do have evidence of a patient who had basically CAR T as a bridge to bone marrow transplant. And that was by far the longest responder in the database that we had.
That's where you see a cure up until 5 years,
yeah,
potentially.
It was 5 years and still counting. And this is, again, this is a kid who's now, up and around for 5 years that basically beforehand had no chance. But and again, that's $1 million therapy treatment.
Yeah.
So that's $1 million regime. Anya, any comment on that so far from your perspective?
Yeah, I can only agree that if you have to make a decision from an HTA perspective, and there is such a discrepancy between what is presented in the submission as the treatment effect coming from a clinical trial that obviously was set up with a certain idea, but then you do realize that the use itself is influenced by not only the results of the trials, but also, for example, the reimbursement landscape in which they are placed. And you are supposed to make this decision, as we always have to, in context to other treatments, and with a societal perspective. Then it feels very uncomfortable, I must say, realizing that you were forced to make a decision based on data that potentially didn't represent the real world situation at all. And I mean, we might come back to this later about the differences between the US and Europe, and how within Europe HTAs assess and how they see this. But in essence, one has to say that it's almost like a drug coming in on a false pretenses, and then you have to make this decision. And like Amar already pointed out, if you make the wrong decision, you cannot reverse it. And here you really have to say this is not a fair playing ground, when you realize that what we do as HTAs will ultimately also affect how the treatment is used in practice. And this is kind of some kind of interaction that we haven't fully understood. But here it's a very brilliant example of where you see how price and availability and all these kind of things actually affect and partially overrule the clinical trial data. So
Correct.
This is a very interesting system.
Yeah, and again, what we see here, and again, this is primarily from US data right now, what we were working with, but what you can see here in the cost, and this is really just adding up every single code that we had in the database. So we're not extrapolating any data at all. We're just actually adding up every single cost code according to Medicare, Medicaid, HC, PCS codes. So this gives us a very accurate picture. What you're seeing is if you just take the internal hospital CAR T costs, this is without the drug, so just the overheads in the hospital to infuse, transfer, get the patient incubated, ready to go, it's about $100,000, about 100,000 euros, all right, which is substantially lower than the cost of a bone marrow transplant. But what that means is the bone marrow unit, that's $300,000 that goes to the bone marrow unit. There's a process there. They're billing that. That's a profit center. It's acting as a profit center. And where if you get to the CAR T therapy, essentially what that means is you're taking $400,000 in revenue that you would normally have coming into your unit and you're transferring it to a company. And I think that's where there's been such a reticence on behalf of a lot of these transplant units to take the CAR T and put it broadly across because they're actually losing money. And in some respects, we've seen some data that they're losing $50,000 to $60,000 of CAR T treatment within the units. So there's a real disincentive right now financially to doing this. And we ran the quality, we did, many of you know me and I'm quite hostile to quality, but we did do some quality calculations on this. And believe it or not, we found that if you get it down cost competitive around $250,000 to $300,000, which is still pretty good. All of a sudden you can get, we think there would be some sense, we did some sensitivity analysis there and we do think there'd be some substitution. And then at that point you'd have some experimentation. So the price maybe needs to come down a little bit. That may happen with competition. It remains to be seen. There are 236 CAR T trials going on right now in the US. There's 31 in Europe. There's a bunch of activity going in the under hospital exemption. We'll have to see what happens. So given the success of our proof of concept and what we just did with the results we've just gone through, we're launching a new real-world evidence project in diffuse large B-cell lymphoma, that's DLBCL lymphoma. And this is going to be funded by the Zorgens Institute. And the idea is we're going to build a historical control for DLBCL, and then we're going to develop an accurate understanding of the treatment pathways of CAR T, both of the current licensed therapies, both in Europe and the US. Hopefully we'll have more data than we did last time. Then we'll look at the pathway and performance and economic costs of the hospital exempted products, assuming we can get the data. That'll be interesting to see if we can pull that off. And then we'll quantify the time for extraction to infusion for all the various products. And then we'll develop a firm understandings of the similarities and differences of the DLBCL treatment pathways between the US and Europe. So we'll quantify those differences. Amer, this is coming from Zin. We're working with you on this. Why do you think this DLBCL is important from the next phase?
I think, well, you can have several reasons. One of them could be a sort of a validation, a secondary validation of the methodology developed to see whether we can keep implementing this in a broader scope of advanced therapy, medicinal products, and or gene therapies, which are coming at us at quite an alarming rate. We do have an extra strength of numbers with DLBCL, and also we're now living in a different time period in the sense that most of the HTA advices have been issued on DLBCL. So now the question also becomes a question of long-term follow-up and what can we do with this kind of real-world evidence right after an HTA advice, and can that re-inform our policy making and our decision making? So these are just a number of reasons why this is quite important and why Zin is very willing to invest in it, and not just Zin, VT as well, Anya on the line, and we have a scientific advisory board of several stakeholders who are very willing to put their thought and time into this.
MEDEV, EMA will be there as well.
EMA.
Yes, it'll be interesting. So, Amer, you're going to talk through some of the situation here from an HTA perspective. The floor is yours. Thank you very much.
Sure. Thank you very much, Dwayne. So basically, this presentation gives a bit of context, and Dwayne, you already mentioned some points, so I won't bother everyone with with too much repetition. I think what we have to highlight as well from not just an HTA perspective, but rather from a health systems perspective, is that new treatment modalities really are coming at us at quite an alarming rate. So this is one example of a publication of a review where they have many different classes of gene therapies lined up and with different details as well as to how far they're on the market. We're also getting requests for scientific advice on new indications, so AML, I think sickle cell anemia, you name it, a lot of gene therapies. I think the challenge is just to reassert them once again. We usually get evidence packages with phase 2 data, phase 1 data, in combination with claims of curation. So you have little evidence and relatively large claims, um, which are not usually not enough to substantiate the claim. The data on hard endpoints, so we have some on relapse-free survival, but overall survival, no data yet. We have to take a go or no go decision as a government body, so we have no options to delay that or wait on extra evidence. And I think also the current effectiveness and cost effectiveness methodology and policy frameworks don't help in providing these different policy methodologies to wait and see or gather more evidence.
That's a quick question then, Amir. How challenging is it, and how much pushback has there been to try and implement change this way, where we're looking proactively at real world evidence?
I think there is a lot of hesitancy, not just from HTA agencies, but also from other stakeholders, so including industry, including regulators. Um, it's very difficult dealing with new questions using new methodologies. I think people are more comfortable dealing with new questions with the
Old methodologies.
old methodologies, which is understandable, of course, because I think from a government perspective, at least what I can say is if we use new methodologies and they lead to very different decisions, we can, not just by industry, but by patient groups and clinicians, be asked very serious questions as to why is the consistency of decision making suddenly very different
Right.
with the new methodologies.
Change is difficult.
Yeah. Just a quick story as well. This is a bit apocryphal. We do have horizon scanning activities, and here I draw a very strange metaphor, but please bear with me. If we look at the healthcare system, it's we're basically looking at an acacia tree, and we have a lot of competitors on the market trying to provide a lot of different kinds of healthcare interventions. So that's the giraffe right there. Basically, this is what trees usually do is that they develop spikes and they develop bitterness in their leaves to try and get giraffes away from them. So that's what's highlighted there. Let's say, very simply put, that's our HCA methods of trying to be very consistent and very thorough in our
So your spikes
package.
on an acacia tree.
We try to position these spikes so as not to do too much damage, but keep everything with good quality and good payability and affordability for the society. But it doesn't seem to work. And I think one question we have to ask ourselves as agencies as well, we have horizon scanning activities. We know which therapies are coming at us and their claims and their associated budget impacts. And can we act on that kind of knowledge? And I think that was one of the very first reasons why we went with the first study on ALL. And just to highlight, we did that study before we even had an evidence package delivered at ZIN to do our actual assessments. So here we go, moving from reactive to proactive HTA. I think the conventional process is rather reactive. We wait for a dossier delivered by a manufacturer. We usually have evidence now with gene therapies, at least, and advanced therapies based on phase 2 trials. We review these dossiers and then we issue an advice to the Minister of Health. And I think the advantage is, of course, we have the luxury and opportunity to critique evidence, but we are not obliged to acquire or develop it. I think people underestimate how much effort it takes to gather evidence. On the other hand, we are dependent, therefore, on several external parties for that evidence on effectiveness and cost effectiveness. And I will make this point, and I believe several of my colleagues at industry will not be happy, but it's not directed against them. But no matter which data is presented, there is some sort of a bias if that data is coming from a particular stakeholder, and we are aware of that. But I'm sure people on the other side of the table can say, "Oh, it's the same thing about HTA agencies. You always go out. You're also biased."
Or hospital exempted trials for that matter as well.
Exactly. So basically, can this kind of real-world evidence inform proactive HTA so we can have an independent analyses and insight into the factors affecting clinical effectiveness and cost effectiveness? We do have to admit, though, we have limited knowledge on how to do so in practice, and I think that's why there's a more of an emphasis on delivering concrete examples where we do that. And just to clarify, with real-world evidence, what we're saying is any kind of health data generated outside the context of randomized controlled trials. Now, if you look at CAR T therapies, Zin has already, as other HTA agencies, issued some reimbursement advice. So you had to the left there the ALL advices, and to the right the DLBCL. What we was something quite interesting. We had two products, Yescarta and Kymriah, and this has led to some um, sensitivities in the media, and we have had a lot of reactions that for one product we've said it has a therapeutic added value. We haven't been able to say something about the cost effectiveness and how reliable the ICER is because of a lot of uncertainties. And we have advised to go through a long-term follow-up of outcomes data through the European bone marrow transplant registries. Whereas for the other product, Kymriah, we said it has no added therapeutic value. And that was quite a strong statement that was due to several limitations in the evidence package, although both products are seen by clinicians as one and the
Interchangeable.
same.
One other thing, though, isn't it true that also any CAR T product that's coming in is a 15-year follow-up, mandatory under the license, but the EMA level?
With the EMA, and I think Anya knows, of course, a lot more about this, but the EMA has asked for post-authorization safety studies on both products.
That are currently licensed for 15 years.
Exactly.
So, Anya,
and just,
go ahead. Yeah, finally.
I think well, just one last slide is we have to realize that we can't give an advice as an HTA agency and say we've lived happily ever after. It is within our remit to follow up on clinical effectiveness and cost effectiveness, to reevaluate products. And I think we need to find the answer to the million-dollar question: how do we best do this as agencies? And I believe that was my one of my last slides. In the sake of time, we could skip that one.
Okay, just to remind everybody, talking about the DLBCL study that we're going to be launching, if any of you out there, there are 100 and some odd of you out there now, would like to participate or are interested, just pop us a note at info@vitaltransformation.com, and we'd love to have your participation. If we can organize it out, we'd love to have you there. So, Anya, you'd mentioned to me over a couple weeks ago that IQWiG had published a study saying that there's an actual need for start harnessing real-world evidence because of the gap that we're getting in data that are coming through in regulatory applications. I just about fell off my chair. Can you walk us through the IQWiG study that was published and your position at NOMA on this issue?
Well, just to come back, yes, it's 15 years for both of those products, but I think it's not written in stone. So once enough data is collected for the initial product, this follow-up period might actually be shorter for newer products that are coming, because if there are no safety signals, then there's no reason to demand 15 years. And it's mainly focused on safety, but I think we see that we would like to see more than just safety data. We would like to see efficacy monitored, and there we have this problem between what can be monitored in real world versus what is usually monitored or considered a relevant primary efficacy endpoint, for example, in a clinical trial. And here in the CAR Ts, I think we did see that there was a problem with how we define our primary endpoint and how clinically meaningful that endpoint really is when it comes to informing treatment decisions. But when it comes to new methods for this comparative effectiveness, the real world evidence, and I don't really like this word because we were accused of that there apparently then was unreal data in the world, which must be then the randomized clinical trials, which are not unreal data, they are just different data. No. So I tend to call it external data, because that's what it really is. It's not generated in a comparative setting, and it's not collected in a randomized clinical trial, but it still is relevant data. And the only thing is that you need to know which question to ask that this data can answer, and understand that it might be different questions than the ones that you answer with a randomized clinical trial. So on the next slide, the question we usually are asked is, why is what is good enough for approval not good enough for reimbursement? This has been the standard question in many drug developments and for many products, while here in this setting on the next slide, the question should be slightly different, because here we are asking ourselves, why is what was good enough for approval not relevant for reimbursement? And that has to do with this fact that I say that sometimes the randomized clinical trial might be focusing on aspects that are different to what might be relevant and interesting to an HTA decision, Also because HTAs do not decide on just the benefit risk as regulators do. HTAs have to decide on the cost effectiveness, and that is a far more complex process to decide whether or not something is cost effective. And on the next slide, you will see that the basic definition is a systematic evaluation of the properties, the effects and or impacts of health technology. And it's the impacts that are not included in a benefit risk decision. And the purpose of this entire exercise of doing a health technology assessment is to actually highlight and understand the direct, the indirect, the intended, but also the unintended benefits and consequences of the adoption of a healthcare technology. And here we can see that in the CAR-T field, we had a couple of unintended benefits that were not clearly the main focus. And we might have some indirect effects that we haven't really taken into consideration, and we couldn't in a randomized clinical trial. So on the next slide, one of the major differences between cost effectiveness analysis and establishing a benefit risk is that cost effectiveness almost always includes some kind of modeling. So we are in essence trying to predict the future. And we have to do that based on the available data from different sources. But they are different not only in type, but also in when they are generated or when they even can be generated. And that's why I tend to say that the regulatory benefit risk decision is more like taking a photo on the left-hand side. At one point in time, you make the decision and you decide whether or not something has a benefit. HTAs are more like running a movie. We need to follow up. We need to predict a little bit of what might be the future. We have to then verify if our predictions were correct. And the latter is usually a bit of a problem, because on the next slide, we see what we are really interested in. So we want to have a shift of the conventional scenario where only RCTs are getting into the equation before the licensing decision, and everything else, everything that's observational, everything that comes from the everyday clinical setting really only comes into the picture after you have already launched your drug. And that means that at that time point, the HTAs also have to make their decision, because the decision time frame is getting tighter and tighter. So HTA submissions are very rapidly submitted after the market access approval is granted by EMA, and that leaves very little time for the HTAs to actually get additional data. And in old days, that wasn't the case. There was quite often enough time after the market access authorization and the actual launch, and before companies were coming to ask for reimbursement, to generate additional data. Nowadays, it's very rarely the case. But we do understand that as HTAs, we would rather have some observational evidence that tells us how a drug actually performs in the wild. To go back to Amr's Africa
Metaphor.
analogies. So there is a savanna out there, there are predators, there are gazelles, and you really want to know how all these different species work together. And it's getting far more complex once you have all the lions and the cheetahs and I do not know what hunting the gazelles, versus the admittingly slightly artificial situation of a randomized clinical trial where you only have one predator and the gazelles in a very confined space. So on the next slide, this is something that probably everybody has by now already seen. The good old-fashioned past is a blue curve where we generated the bulk of evidence in the development phase. Once we launch, evidence generation rapidly decreases and usually was focusing on safety. Nowadays, there's lots of information that can be only actually properly collected in the post-launch phase. But this is not available when we have to make our decisions. And that is probably our biggest problem at the moment, because on the next slide, it's not just that we have huge differences, for example, between the States and Europe. It's very clear from your example that the price actually affects the behavior of the physician. in Europe, obviously, between the physician and how he might be affected, he or she, there is another group of people. There are the HTAs and the payers. They actually already come into an equation, and they might change the picture completely. They might enforce sticking to the indication wording far more than maybe in the States. But then, to make it even more complex, on the bottom left, you see the European system. You see that every country has at least one HTA. Most of them have several agencies that are splitting up tasks between them. So there are a lot of people that are sitting around the table, and if they all take the data as it is, then as a matter of fact, it might only be relevant information for one of those, because the context is what makes the difference here. So if IQWiG reports that they had less than 50% of their drugs assessed as having an additional benefit, then that is because they put it without modeling, just purely on contextualization of the entire healthcare system, meaning that a CAR T cell has to compete with any other available treatment, and they would be extremely interested in the type of data that you have provided, because for them that's the only type of evidence that can help them make this decision. For people like Amra, me, or the people from NICE, it's a bit easier because we can at least do health cost utility analysis. So we have health economic modeling that can help us, and that allows us to put into place into those models different scenario where we can choose to, for example, decide that, well, maybe it is more a bridging therapy and not a intended cure as the company wants to convince us of. But that is only possible if you are willing to model. So the IQWiG picture is maybe a very dark one, where you see that they have a huge problem finding additional benefit for new drugs in many cases, because they cannot really see the benefit when they put it into context. And this is where I earlier said it is really not just the photo situation we have. We have to realize that there are unintended impacts that getting a new drug on the market can have. And the IQWiG is very conservative when it comes to this. I think that the CAR Ts are an example where most HTAs actually were rather proactive, which is a good thing. We have all seen what was coming. We were not as surprised as we used to be in the past, but it still doesn't solve the problem that the real world evidence is not the solution to everything. The way you used it here in your publication is a very good one. This is exactly how I see that the big value of real world evidence actually can be harnessed. The lesser pleasant version of the use of the real world evidence is how most of the companies were arguing how they would establish relative effectiveness by trying to convince us that single arm trials are working here, and that you can use this kind of data to construct some kind of contextualization or almost an artificial control arm. And that is where real world evidence really isn't as good as many people would want it to be, but experience tells us it isn't, because it's two different types of beasts. And that's where I feel that you have provided a very good example on how this can be very useful from an HTA perspective. The problem lies in the timing. So the data is only coming once the drug is already approved. It has to be approved, because otherwise we cannot generate the data. And there it still is a bit of a chicken egg problem, I think.
Well, that's why we were very fortunate where we were able to tuck into a clinical trial site that had a lot of various clinical research studies. One quick question for you, Anja, related to your presentation here. And again, thank you very much. I was at least heartened to hear that IQWiG, which has been so against real world evidence, is now saying, look, you know, we got a problem here. I mean, isn't identifying the problem at least part of finding a solution? I mean, isn't that a good thing?
Yes, they actually have just changed the legislation in Germany, and they are now having a legal framework to request post-launch evidence generation. So it's very clear that also IQWiC has understood that there is on occasion no other way forward.
Yeah.
That still doesn't solve the problem that as long as we have the situation as it is nowadays, that once the submission comes, we have a certain time frame, we have to make a decision at the end. We have to advise someone to say yes or no. And if we don't have the relevant evidence, then the first step forward really is that all HTAs embrace this concept of saying we have to build in some kind of flexibility into the system where we say, well, maybe we don't pay the full price in the beginning, but if after a couple of X years you can prove us something in the magnitude of, and that magnitude of needs to be agreed on. And that can only be agreed on if we have a firm understanding on the true effect of the comparators in that particular patient population. And then we can start discussing on what a realistic target would be to provide within a certain time frame by the company based on observational data. And then really understand that these observational data have to stand alone. They are not some kind of replication of the randomized trial or whatever. They have to come self-standing, because it's very clear that you can, for example, establish a benefit risk based on an endpoint like event-free survival. But event-free survival for HTAs could be subcategories of utmost importance, because one of these events might be a very costly event, another event might be rather cheap. And you've shown that. So the stem cell transplantation is on the first, in the first instant, it seems more expensive, but it might in the long run not be. And it might be a preferable outcome for the patient. And again, event-free or progression-free survival, those are endpoints that are actually meant to buy us time, in the sense of that we decide earlier on that there is going to be some long-term effect of a certain magnitude, but from the HTA side, we need to have some insurance about that magnitude at some point. But at some point really means that we have to learn to understand that that point is not the market access authorization, because likely the evidence package will be too limited at that time point. So yes, we need some kind of change in our system, a certain willingness to accept that we have to make contracts with the industry. We pay you this much. If you succeed, you get more, you get the full price, or you get an extension or whatever. But that is the way forward, because otherwise we are still stuck with this idea that, okay, how big is the mistake if the drug doesn't really work as well as they say it right now?
Anya,
and then it might stop something that actually is very promising.
Anya, I agree with you. I think the problem from an industry standpoint is when I think the industry would 100% agree if it works well and the price goes up. I just don't think anyone's ever seen a price go up. I think that's part of the problem. And I think the industry gets cold feet because they know that generally the prices only go down. So I think if we can put something in place where if that's serious, if things do go up, if they perform better or at least hit the targets, I think a lot of people would agree with that.
And I think also just to add, industry is also taking a part of its responsibility in trying to think of new
methodology,
models and new methods. So it's not a good versus evil, a black versus white. It's always much more nuanced, and I think it's gonna take all of that collaboration together to try and reach that middle ground.
So we've got about, thank you, Anya, again, really great. We've got about 15 minutes here, and we've got a bunch of questions that have come in. Anita Burrell, yes, thank you for commenting that we are only showing the presentation version. If you knew the scramble we had to get live today, I think you'd probably understand that we're just glad to be live, but I appreciate your comment. We will do better next time, I promise. Konstantinos Likopoulos, can the speaker please correct that PALL? There's only Kymriah, the Novartis product. Yeah, Gilead is only in DLBCL. That's correct. I just should clarify that there were multiple clinical trials before the license in several products. There were several trials that were canceled. I don't want to go into names. And Vanderbilt was a clinical trial site for the class. Multiple clinical trials came in. We did not bias to a selected licensed product. We only looked at the class. So yes, while there are 2 licensed products, only one of which is for ALL, there were several trials ongoing. I don't want to dig more into that because that violates some of the confidentiality of what we have, but I hope you can read between the lines there. There were a bunch of trials going on, and so some of the data is for the class. So again, we should, and we do say this quite clearly in the paper, it is not indicative of a particular product, it is indicative of the performance of the class and the treatment approach to the class. I hope that's clear. Thank you. Paul Robinson, surely this means we need a head-to-head CAR T versus HCT. Couldn't agree more. Amir, what do you think?
No, I certainly agree too. And while writing the assessment reports, I think I found one trial where this is being trialed. I think this is Zuma 9 or Zuma 8 where this is being done. Definitely interested in the outcomes and waiting to see whether they correlate to some extent with what we find.
Yeah, and Alan also commented, yeah, we all, I think we're all in violent agreement there, folks. Anya, what do you think? Should we be going that way?
Absolutely, but I think that's where I say that the original plan was excluding patients that were relevant for stem cell transplant.
Exactly.
So that's where you cannot complain that the company set out with a certain intention, assuming what that drug would do. Now it's very clear we should challenge the industry and saying, well, look, you need to broaden your horizon when it comes to how you want to test it, and in certain circumstances, head-to-head is really the only way to go, and we have to maybe send out a much stronger signal saying that this endless discussion on why you cannot have these kind of trials and why only single arm, we really have to argue that we can use thousands of patients in trials that do not answer any question. If we would have used only 200 of them in a really relevant trial, we would be in a much better place.
And I think with a certain level of data granularity, Anya.
Yeah.
Yes, absolutely.
But I will say this. I mean, no one. Let me rephrase this. I had a meeting with a high-level HEOR office of a company that's currently working on CAR T. I'm not going to go into details. But when I told them that clinical practice is evolving to use CAR T as a combo therapy, as a bridge to bone marrow, they looked at me like I was crazy. Well, why are you going to do that? You kill all the CAR T. I'm like, well, the CAR T are dying after 6 months anyway. They're losing their efficacy. So this actually gets you a longer efficacy curve. I think a lot of the assumptions on you that were, again, what we saw with some of the curative aspects of CAR T in the less disease burden population, I think there was a lot of media that went into this, but then when the rubber hit the road and the practitioners were only putting it in the sickest of the sick, it was like, oops, this isn't working like we thought. So I don't think there's necessarily been some sort of mendacious, black cape, tying a woman to the railroad tracks here. I think it was just people got very excited, but then the clinical practice has changed a bit. I mean, can you at least see that?
Sure. I think it's really, we need to understand that this is like, again, the savannah. We are part of this environment. So what we do, any action from our side has also reactions. And the same goes for the industry, and the same goes for politicians. So the moment, for example, the prices are high and you have responsibility for the budget of your hospital, for example, you react very differently versus in the European situation where these decisions are made by someone else and you as a physician do not necessarily have to think about this. So that in itself might actually very much influence how the CAR Ts are used in practice. And that might differ not only between the States and Europe, but also in the different countries. It's obviously that a very experienced transplant center that has very good results reacts differently to this kind of treatment than a center that actually has very rarely does a transplant, has a not so good track record, does realize the risks are much higher than using CAR T. It's logical that there will be regional impacts on how drugs are going to be used, and in particular these kind of technologies, how they will be used. And that in itself is something that we are also steering, not necessarily directly, but indirectly in how we behave and the way we also describe and how we reimburse and how we communicate our decisions.
If I could just add too, Vanderbilt University is ranked, depending on the year, 1st to 5th, somewhere in the top 5 on an annual. They're considered one of the best stem cell units in the United States. Now that said, they don't I've had some very cynical practitioners here in Europe say, well, obviously they're biasing who they take in. No, they don't. We looked at the data, we analyzed the data, and they're taking everybody. So they're not just only taking the easy ones, they're taking the hard ones too. So they're just a very good unit. So does that have an impact on the data? Perhaps.
It could.
Mhm.
It could have some sort of impact. Not to go in on quality differences or outcomes between different stem cell units. But I think also we had that discussion before during that project. If anything, it would bias the difference in a certain. I think we had that discussion about comparative effectiveness,
Sure.
and that it would actually, regardless of how that would bias the outcome, it still doesn't stand next to the general bigger question of, are these CAR T therapies curative
Sure.
in practice? So it's an important point, but I think it may be a nuance that distorts the
A
discussion as
distinction
well.
without a difference. I guess.
Yeah.
We've got a
And
lot of.
anyhow, I mean, bias is not something people are usually very afraid. They consider it something extremely negative, which isn't true. Bias is just simply a fact of life. You need to understand it, and then you can still interpret the data. So that's what you did.
And
So
speaking of bias here, Anya, we have a question from Eva Malikova. With regards to the analysis of RFS between CAR T and HCT, isn't there a bias? Thank you. Since you have double the number of patients in the ACT group. Yes, obviously. Now we did run, just to be very, very clear, the Kaplan-Meier curve does account for bias in the patient population, and we did do a log rank test to do a comparator head-to-head between the two populations in the context of the Kaplan-Meier curve, which does account for a right skew. So we did manage that as much as we could. Obviously, is there a potential bias? Well, we did find the bias, and it was in the treatment selection. So yes, there was a bias in that the patients getting CAR T are sicker. Basically, that's so. Yes, that's what we were trying to figure out: is why was the bias? There was the treatment bias affecting the outcomes, and yeah, that was the case. Cecilia Young, will you distribute the slides deck during the presentation after the meeting? Well, actually, I can do better than that. If you go to the BMJ, Evidence-Based Medicine, you can actually just download the study and everything's there. So you can, it's open access. So please just go type in, Zin, Amir, Dwayne, type in CAR T and Vital Transformation, and you should just get right to you can also go to our website, vitaltransformation.com. There's a link right there, and you can download the actual study and show it around to all your friends for free. So our gift to you. Thank you for participating, and thank you for the question, Cecilia. Let's see, from Ilfeng Liu. From my understanding, the objective of the study is to assess whether CAR T does win in the real world in contrast to two referred phase 2 studies, reference 2 and 3. Would the authors be confident to include a solid, narrow answer in the present study? Okay, I don't know if it's a win-lose scenario. I think it's sort of, this was much more, and again, Anya, you might want to, and Amir as well, does it do what it says on the tin? I think that was really, is what we're getting, are we getting what we're buying? I think that was really the question. It's like, what's really going on? And I think what we saw is that the studies that are presented by ICER were not necessarily representative of how it's being done in actual clinical practice. Now, again, I don't think there's any maliciousness there on behalf of the practitioners. I think they're behaving in their own best interest. But what we were seeing is clinical practice is not the same as what was presented in the ICER studies for evidence. Amr, any comment on that?
Yeah, I think you hit the nail on the head. Is basically we have an advanced therapeutic medicine product with a claim cure. We were trying to say, before we even get to an evidence package being submitted to us, what can we say about this product in the sense of how is it being used in practice? What are the effects that are coming out? Is it curative or not? I wouldn't say win versus lose versus, because then we need explicitly a study that looks only into that, and I don't know if we had the data set to do that. So that's where the context comes in. Perhaps with a DLBCL study, because we're now in a different phase, it's post HCA advice, we do have some use in practice, etc., that we can go into this comparative question using methodologies such as match adjusted comparisons, etc. So that's a brief answer to that.
Anya, last comment to you. Any other comment there to add?
No, I don't think it's that the can says something different than what you get. It's what you see with any drug in its life cycle. Its use changes in time. It's getting more fine-tuned. Physicians do understand better what side effects are there and how to handle it properly. I think nowadays just everything happens far more accelerated than it used to be like 30 years ago, and it's more obvious. Um, the 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 thing that we need to achieve, and that is what you are doing, is also to some extent understand how much more data do we actually need to make a robust assessment of the reality. So do we really need 15 years of follow-up, or is 1 year follow-up once the drug is released actually going to be enough? Because then we can start discussing with the industry what kind of commitments are required for a more flexible reimbursement process.
Well, in our case, Anya, it was 3 years of retrospective data. We didn't even need to look forward. We could look back under the clinical trial regimes. Again, we didn't get clinical trial data. We just captured the patients in the EHR and saw how they did, and we're able to make an assessment. So it doesn't necessarily need to be forward. With that, we've gone over by a couple minutes. I'm sorry about that. There were 110 of you on the webinar. Thank you very much for your participation. There are 6 questions unanswered. Amir and Anya and I will bat those around. We will answer those 6 questions and send you responses via email. Anya, thank you very much for your participation. I know you had a heck of a night last night with tornadoes and lightning and power outages, and thank you very much for being the trooper and being on board.
Thanks. It's always a pleasure.
And again, Amir, thank you very much for your hospitality.
My pleasure.
And thank you all of you for being on the webinar. We look forward to the next study. Again, vitaltransformation.com. You can download the study for free, the ALL study, and we'll be in contact with you all in a year from now when we have the DLBC results. Thank you very much for your time.